Hello, students. Various scientists invented various aspects of molecular genetics, structural and functional aspects of genes have also been identified. Now, we thought about how genes can be useful to mankind. As a result of it, the Human Genome Project was founded in 1990. Let us see it in detail. The Human Genome Project is an international scientific research project. The primary goal of the project is of determining the sequence chemical base pairs which make up DNA and identifying and mapping the approximately 20,000 to 25,000 genes of the human genome. In 1990, the project was initiated by the joint efforts of the US Department of Energy and the National Institute of Health. A working draft of the genome was announced in 2000 2001 February. The analysis of the working draft was published. Now we'll see the aims or the goal of the project to prepare a genetic map of all the genes in the human beings in full detail. So the information of nucleotide sequence of each gene and its mode of expression should be determined and stored. Then to store the information in databases and improve the project's tool for data analysis and transfer related technologies to the private sector to understand social, ethical, and legal complications related to the project and find out proper solutions for them. These were the basic goals of the project. In April 2003, the Human Genome Project sequencing was completed, and the human genome contains 3 billion chemical nucleotide bases. The average gene consists of 3,000 bases. Now we'll see the methodologies, the methods involved two major approaches. The first one, identifying all the genes that expressed as RNA, expressed sequence tags ESD, and the second is the blind approach of simply sequencing the whole set of genome that contained all the coding and non-coding sequences. Later, assigning different regions in the sequence with the functions, that is the sequence annotation. Now let's see the salient features of the human genome. Some of the observations drawn from the Human Genome Project was the human genome contains 3 billion nucleotide chain bases. The average gene consists of 3,000 bases, and the total number of genes is estimated at 30,000. Almost all nucleotide bases are exactly the same in all people. The functions are unknown for over 50% of the discovered genes. Less than 2% of the genome codes for proteins. Repeated sequences make up a very large portion of the human genome. Chromosome number 1 has most genes, that is 2968, and the Y chromosome has the fewest, that is 231 chromosomes. Sorry, genes. 231 genes. It is expected that we will soon have snapshots of more than 1200 genes that are responsible for common cardiovascular ailments, endocrine diseases like diabetes, neurological disorders like Alzheimer's, cancer. The human genome sequencing not only holds promise for a healthier living but also holds the prospects of a vast database of knowledge about designer drugs, genetically modified diets, and finally, our genetic identity. Scientists have identified about 1.4 million locations where single base DNA differences, SNPs, are found. And this helps to find chromosomal locations for the disease associated sequences. Thereby, we can trace human history. One of the greatest impacts of having the human genome sequencing may be enabling a radically new approach to the biological research work. In the past, researchers studied one or a few genes at a time. And now, with whole genome sequences and new high uh, through, throughput technologies, we can approach questions systematically and on a much broader scale. So, deriving meaningful knowledge from the DNA sequences will define research through the coming decades, leading to our understanding of biological systems. So, for that, we need the expertise and the creativity of tens of thousands of scientists from various disciplines in both the public and private sectors worldwide. I will see the applications of human genome project in molecular medicine. It improves diagnosis of diseases and is also used in gene therapy. It helps to improve the understanding of diseases like Alzheimer's disease, Parkinson's disease. Then, with the help of Human Genome Project, we can rapidly detect and treat the pathogens in clinical practice, develop new energy sources, and monitor environment to detect the pollutants. This is used to evaluate the health risks faced by individuals who may be exposed to radiation and to cancer causing chemicals and various toxins. This Human Genome Project can help to identify potential suspects whose DNA may match the evidence left at the crime scenes to establish paternity and other family relationships. Uh, this Human Genome Project is used to develop biopesticides, incorporate edible vaccines into food products. The project's goal included not only identifying all the genes in the human genome, but also addressing ethical, legal, and social issues. The human genomics will provide a healthy and disease free life. It is expected that we will soon have snapshots of more than 1200 genes that are responsible for common cardiovascular diseases, endocrine diseases, and all. Like the fingerprinting that came into use by detectives and police labs and others during the 1930s. Due to its specification, each person has a unique DNA fingerprint. Unlike a conventional fingerprint that occurs only on the fingertips, a DNA fingerprint is the same for every cell, tissue, and organ of a person. DNA fingerprint cannot be altered by any treatment, but fingerprint can be changed by surgery. So the DNA fingerprinting is rapidly becoming the primary method for identifying and distinguishing individual human beings. An additional benefit of DNA fingerprint technology is the diagnosis of inherited disorders in adults, children, and unborn babies. Even blood-stained clothing from Abraham Lincoln has been analyzed for evidence of a genetic disorder called Marfan's syndrome. DNA fingerprinting technology was first developed by Alec Jeffreys in 1985. And this is the technology used for identification of individual at genetic level. DNA fingerprinting involves identifying differences in some specific regions in the DNA sequence, which are called as the repetitive DNA. So, a small stretch of DNA is repeated many times. These repetitive DNA are separated from the bulk genomic DNA during the density gradient centrifugation, by which you can separate this. The bulk DNA forms a major peak, and the other small peaks are called as the satellite DNA. Now, depending on the base composition, length of the segment, and number of repetitive units, the satellite DNA is grouped into many categories like the microsatellites, mini satellites, like. And these sequences normally do not code for any proteins. And these sequences show high degree of polymorphism, and they form the basis of DNA fingerprinting. Since DNA from every tissue from an individual show the same degree of polymorphism, they become very useful identification tool in forensic applications. These polymorphisms are inheritable. So DNA fingerprinting is the basis of paternity testing in case of disputes. The information contained in the DNA is the sequence of the base pairs or the letters, And in the same way, you can living organisms that look different and have different characteristics also have different DNA sequences, and the DNA fingerprint is a very quick way to compare the DNA sequences of any two living organisms. There are 23 pairs of human chromosomes with more than 3 billion uh, genome size. And not all the segments of DNA code for the protein. Some DNA segments have the regulatory function, while others are intervening sequences or the introns, and some are repeated DNA sequences. For DNA fingerprinting, short repetitive nucleotide sequences which are specific for a person are important. These nucleotide sequences are known as the variable number tandem repeats (VNTR), and this was initially developed by Alec Jeffreys, and he used satellite DNA as probe that showed a very high degree of polymorphism. The techniques involved southern blot hybridization using radio labeled VNTR as a probe. It included the isolation of the DNA, that is, extraction of DNA from the sample cells. Then, restriction digestion DNA is cut into sections by using restriction endonucleases. Then, separation of the DNA fragments by electrophoresis. The DNA band pattern is then transferred to a nylon membrane or nitrocellulose. Then a radioactive DNA probe is introduced, and the DNA probe binds to specific DNA sequence on the nylon membrane. Now then, any DNA which is not attached to the probe is washed off. Then the radioactive DNA pattern is transferred to X-ray film by direct exposure. When developed, the X-ray film reveals the unique pattern of dark and light bands. The DNA fingerprints, when it is developed, it will show like this. You can compare radiography is done to detect the DNA fragments. I will study the application of DNA fingerprinting technology. DNA fingerprints are useful in several areas of society. They are used by professionals in human health and justice system. It can be used to help identify individuals who may have carried out crimes such as rape, or settling paternity disputes, deterring relationships in inherited diseases, and monitoring bone marrow transplants. DNA fingerprint technology is used to diagnose inherited diseases like cystic fibrosis, hemophilia, Huntington's disease, Alzheimer's disease, sickle cell anemia, thalassemia, and many other diseases.